Microsporangium. It is a structure in the male reproductive organ where the development of pollen takes place. Development of anther. The young anther is represented by a group of parenchymatous cells. It soon becomes four-lobed. In each four lobes, some of the hypodermal cells begin to act as archisporial cell. Each archisporial initial divides into outer primary parietal cell and inner sporogenous cell. The primary parietal cell divides to form 3 to 5 layers, namely endothecium, middle layers and tapetum. The primary sporogenous cells divide to produce a mass of sporogenous cells or microsporocytes. These sporogenous cells undergo a few mitotic divisions and form microspore mother cells. The anther wall consists of 4 layers, namely epidermis, endothecium, middle layers and tapetum. Epidermis is the outermost layer. It is only one cell in thickness. Endothecium, this layer is situated below epidermis. It is a single layer of radially elongated cells with fibrous thickening. Because of its fibrous thickening, the endothecium is also called fibrous layer. In between these cells, a few cells without thickening are also present. These thin-walled cells collectively form the stomium or the line of dehiscence. The middle layers. The middle layers consist of 3 to 4 layers of thin-walled cells. These layers are situated below endothecium. These layers generally degenerate to provide nourishment to the growing mother cells. Tapetum. It is the innermost nourishing layer of anther present below the middle layer. It is usually single layered and rich in reserve food material. It is polyploidy and serves to provide food for the developing sporogenous tissue, that is the microspores. The tapetum, on the basis of its behavior, is classified into two types, namely amoeboid tapetum and secretory tapetum. Secretory, or it is also known as a glandular tapetum. It is also known as the parietal tapetum. In this type, the cells remain as such in their original position throughout the microspore development. These cells secrete nutrient materials which are given to the developing spores. It is more common in angiosperms. In the initial stages of development, the cells of the glandular tapetum contain small bodies called proeubish bodies, which are involved in external thickening of the exine of the spore wall. Amoeboid tapetum. It is also known as invasive or periplasmodial tapetum. In amoeboid tapetum, the inner and radial walls of the tapetal cells break down and the protoplast become a plasmodial mass called tapetal periplasmodium, in which the pollen mother cells are suspended. It is found in the anthers of Typha, Tradescantia, Helianthus, Alisma, etc. Function of tapetum. The function is secretion of the enzyme callase to dissolve the callosic wall of tetrads and set them free. The second function is secretion of polysaccharides into the locules during the free microspore stage, which are absorbed by the microspores, that is, nourishing the developing pollen grain. Next is formation of eubish bodies. Next, formation of pollen kit and tryphin, which are deposited on the pollen surface and help to bind pollen grains together and for efficient insect pollination. Sporogenous tissue. It is located in the center of each microsporangium in young anther. With the development of anther, the sporogenous cells undergo meiotic division to form 4 microspores. Tetrad. Each sporogenous cell is known as pollen mother cell. The anther matures and releases the pollen grain.